All right, let's talk about the blood typing. The blood typing, sometimes it's confusing for students, but let's talk about the blood typing. Universal donor, universal acceptor, ABO blood type, and RH factor, positive or negative blood type. So with blood, right, we have three types of blood, or four types. It depends how you look at that. Right, we have A, B, O, and combination of B, A, B. So there's also O is blood type 1, A blood type 2, B blood type 3, and AB blood type 4. You can see this actually on the military uniform. You see the soldiers, they have AB, ABO, and they also have those Roman letters, right? But this has nothing to do with our lecture. So what these letters represents? So this letter represents an antigen. Antigen is just a molecule that's sitting on top of the cell membrane. So let's say that has this particular shape. And we decided to call this antigen A. So and this is the red blood cell. A lot of cells, they have their antigens. Antigen is like an antenna, like recognition. For blood type B, antigen that is sitting on the cell looks like this, right? So this is the B blood. So another blood type has antigen A and has antigen B, right? A combination of these two, like this, right? And O do not have any antigen. But what is an antigen? In human bodies, antigens is a substance that is able to stimulate the production of antibody. Antibody is something that can attack the pathogen or the virus. Why? Because let's say virus, or let's say pathogens, right? That's a pathogen that wants to enter our system, also have antigen, like that. What is antigen? Let's say it's like a flag that says self, meaning that I represent your system. Or it's a hypothetical flag. You can say non-self, that do not represent my system. So when the bacteria or the antigen that has a non-self enters my system, that antigen, there is a bacterial non-self antigen, can stimulate the production of antibody, because my system has never seen this before. And it's been designed to attack and destroy. It's more complicated than that, but I just wanted to quickly explain what the antigens are. So the antigens are these chemicals that's sitting on top of our cells, and they identify our cells. When we do tissue transfusion, transplantation from one person to another person, we want to match as many antigens as possible. Let's say they have different shapes. Red blood cells, they have like 40 plus different types of antigens, but when it comes to blood transfusion or to identify the type blood, we're only concerned about A, B, O, and there's also another antigen. There is RH factor. RH factor positive means antigen D, and RH factor negative do not have any antigens. There's no D. That's basically, it's just with positive, we have that extra antigen. So you can have A positive, meaning you have antigen A and you have antigen B, D, or you can have A negative that you only have antigen A, but there is no antigen D. Antigen is something, right? Antigen is the substance that can generate the production of antibody. With blood type A, you have something called anti-B, that against the B blood. So when you try to bring the B blood into A blood, anti-B will attack B. The red blood cell would be destroyed and will release hemoglobin molecules that transports oxygen, and that can lead to basically failure of an organ like kidney, for example, because the hemoglobin would be very toxic for the system. So B has anti-A. If you try to bring A into B, anti-A will attack blood type A. Blood type AB do not have any antibodies, right? Well, against these two antigens. You do not have anti-A, you do not have anti-B, because if you would have anti-A, you would destroy A. If you would have anti-B, you would destroy B. For that reason, blood type AB do not have any antibody A or antibody B. Blood type O has anti-A and has anti-B, anti-A, right? So there's no antigen A, there's no antigen B, so these two antibodies cannot destroy just because it does not have any antigens. That's basically the structure, what you need to know for now, for the blood type. But there's also Rh, right? Positive or negative. So if you're positive, right, you have this extra antigen, D. If you're negative, you do not have it. So it means that if a person, let's talk about blood transfusion. If a person is A positive and another is A positive, we can safely bring blood here or bring blood there. We can do the blood transfusion. If you're A positive and you are A negative, you cannot bring A into A negative because negative do not have D and have never seen it before. But if you bring this D, which is Rh positive, right, your body will be sensitized, meaning your body will create antibodies against D. And then with the second exposure, that could be problematic. Let's say that during pregnancy, mother has a baby which is Rh positive, but mother is Rh negative. So the baby has this D antigen. And its possibility during delivery or through placenta, that can end up in the mother's blood. Mother's blood has never seen D antigen before, right? The D comes in here. The body, right, will produce antibody against D. Why? Because antigen can stimulate the production of antibodies. If the body has never seen it before, it will stimulate production of antibodies. Then when the baby is gone, everything is fine. With the second exposure, when there's a second baby, there's also positive, and the blood again will appear in the mother's system. So that can lead to may lead to death of a child or the death of a mother. So when you have a first exposure, your body has been sensitized, so it's kind of triggered, and the antibodies has been produced. So the second exposure could be very problematic. Let's talk about a blood transfusion here. Let's talk about the universal donor and universal recipient. Blood type O is being considered as universal donor. Blood type AB is considered a universal recipient. So, blood type O do not have any antigens, right? So for that reason, we can bring it into the AB. There are no antigens, right? There are no antibodies to attack. You can bring O. There are no antigens. Antibody cannot attack. You can bring here. There are no antigens. Antibody cannot attack, right? But what about these antibodies? When we say blood transfusion, do you really think we transfuse the entire blood? So here is the column of blood. Hypothetically, I'm going to break it half and half. One half, we have formed elements. And another, we have plasma. Plasma is mostly water, some proteins, some other constituents. Form elements, that's the red blood cells, right? So there's white blood cells, there's platelets. And here, within the plasma, we also have antibody, plus antibody. So when we say universal donor, and the blood transfusion happens, we'll try to remove antibodies. So when we're doing this, we remove plasma with antibodies. So when they do simple blood transfusion, they try to remove plasma that has antibody and just give you deformed elements, which is a red blood cells, which is this. So for that reason, when we say that O is a universal donor, you're donating that without antibodies. Otherwise, if you would bring the entire blood into here, right, into B, so this B antibody will attack A. This A antibody, if we bring it here, will attack B, A. Obviously, if you bring it here, this antibody will attack A and B. So the universal donor, but there's more to this. The universal donor should be also negative, because if you will give O positive, that's an antigen, right? That's a D antigen. If it's A positive, it's fine. You're bringing D and A plus seen D before. Same thing with B plus, same thing with AB plus. But if they're negative, right, they have never seen D before, and that can lead to sensitization, and that can lead to exposure of the first reaction, production of antibodies, and then with the second exposure, let's say, or even with this exposure, if you stayed long enough on the blood transfusion, that can be deadly. So AB is a universal acceptor. And remember, the blood donation happens without the plasma. But if you're doing A plus to A plus, you can bring the entire blood. If you're doing A minus to A minus, B plus to B plus, B minus to B minus, AB plus to AB plus, AB minus to AB minus, O minus to, right? But if there are no other options, right, so we're using this combination. So the universal recipient can receive O, because O do not have any antigens and do not have any antibodies, right? But when you're receiving A, also should not come with antibody, because the antibody is present in the plasma. So you're just bringing A. A matches A, and you're fine. But B, what about B? Then you can bring B. So that's why this is a universal acceptor. This is a universal donor. If you remove plasma, and within the plasma you have these antibodies. I don't think that this material is complicated. You just have to sit down and kind of play around with that, because the questions on the test would be, what if this person has positive or negative? Is it possible to transfuse blah, 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 right?